Good morning, everyone, or good afternoon, depending where you are. I would like to thank the organizers for giving me this opportunity to talk to you today about choices for genetic testing, in particular about using targeted resequencing. And I'll focus on two platforms, microarray and next-generation sequencing. This presentation is eligible for continuing education credits, and there will be a link at the end of it for those that want to get some points for that. I also want to mention that there is questions and answers that you can post as I continue presentation, so you don't have to wait till the end, but I will be answering them at the end of the presentation. So what I'm going to talk today about is firstly the learning objectives for those that want to get credit. Later, what is really resequencing, why and when do we use it. I'll talk about the technologies used and also about how to go about establishing a resequencing assay in the lab. So the learning objectives are firstly to understand how targeted resequencing is applied to genetic screening or genetic testing, and also to appreciate the complexity of the validation protocols for the resequencing assays. What is resequencing? It's basically a sequencing of part or whole genome of an individual and comparing this to a reference genome for this particular species. In our case, we'll be talking about human genetics, so the human genome. So when do we use resequencing? We use it for a number of applications. And actually, when you think about it, Sanger sequencing really was resequencing as well, since you needed a sequence to first design the primers and then compare your result to that. When do we use it? We use it for carrier, prenatal, or newborn screening. We use it for diagnosing previously undiagnosed childhood or adulthood diseases or disorders. We use it also for risk assessment for complex diseases. And we have a number of different methods. We have panels of genes, exome sequencing, or whole genome sequencing. We can use resequencing for genotyping, for example, carrier screening, or would be genotyping. We have a set of mutations that we want to screen for. But risk assessment would be more of a fishing expedition, thus it will be variation screening. We generally use SNP arrays for variation screening, but whole genome sequencing is becoming more and more popular, especially with now 100,000 genome projects. So not 1,000 genomes, we increasing that number. How do we perform targeted resequencing? There are two main ways of doing this. First is to actually use capture of your target DNA. The second is through PCR amplification using specific primers. So what's the difference? When you're capturing, you need to design probes. And there are lots of commercial probes that you can use for this. And they are either done in solution when the probes are linked to beads or on arrays. And in this way, after the DNA has been fragmented, you can basically pull out what you're interested in. PCR amplification, on the other hand, there is a number of different ways of doing it. The simplest is just multiplex PCR. You can use also Raindance, Fluidigm, Haloplex, or any of the proprietary systems from any of the companies. The main difference here is that the primers that you design have to be screened for SNPs and repeat sequences, so to ensure that you can actually amplify cleanly what you need to amplify. Now, when we think about genetic testing, if we go back 10 or 15 years ago, we all used, for sequencing purposes, we all used Sanger, and this was kind of a little slow, one gene at a time or one exon at a time. And if your genes were large, that it took quite a long time, and it was quite expensive. And then as we started sequencing more and more genes for our testing purposes, it became obvious that Sanger is really, really expensive and it's time-consuming. So for sequencing that varies from 1 to about, say, 10, 20, maybe even up to 40, that's what I did. You can use microarrays. However, when you move past hundreds into thousands of genes, the only way to go is with next generation sequencing. So our sequencing becomes more complex from the low complexity of Sanger to really super high with next generation. Now, there are a number of assays available. I did put in this table Sanger sequencing because there are ABA resequencing sets, and if your lab is doing a lot of single gene sequencing, the same over and over again, that might be a good choice for you to use one of those. When it comes to microarrays, there is really only one commercial panel right now, the pan-ethnic carrier screening panel. The 3 previously existing panels have been moved now to next generation sequencing. And when you look at the next generation sequencing and trying to bring this into the clinic, you will see that there is a number of assays possible. There is carrier screening, also a carrier screening panel, but there's a number of others: Noonan syndrome, cardiac disease, pharmacogenomics, neurodegenerative. There's multiple cancer panels. And you basically can pick and choose, and you can design your custom ones as well. Now I'll go into technical comparison of the 2 platforms. So just to start off with the sequencing microarrays, what they are and how they work. The sequencing microarrays are based on the Affymetrix technology. So a lot of you are familiar with it, and those that aren't, I'll explain a little bit more about it. Those arrays come currently in 2 different formats, really. One is the traditional cartridge form in 3 different sizes, from 49 to 169 format, allowing you to sequence anything between 47 to over 300 kilobases. More recently, the strips of 4 chips have been proven to actually work and support resequencing. And those, as well as the 96-well plates of chips, actually require different equipment than the traditional cartridge variety. So there are some choices that you have to make if you decide to go this way as to what your throughput's going to be. Now, very important part is how actually does the resequencing work? I had this question asked a lot of times, and I think something happened here because this is looking a little strange. I was looking fine yesterday. Basically, all of the Affymetrix probes are 25 mers. And the way it works is that if you are going to sequence from this C here that I'm pointing with my arrow, you have to have the first 12 bases added to the sequence from the gene, from your target gene, and those will not be ever read by the system because the AFI requires the first 12 bases as a buffer, and it starts reading the very first is the 13th base, and the 13th base is the one that's variable, and you can have T, G, C, or A in that position. If you look here, we have a G in our reference genome and an A in a patient sample. And this line got completely out of whack here. That G should be under the A. And basically what happens is that you read along the sequence. So if you imagine having your reference sequence tiled in 25 mers underneath, and the 13th base is always the one read, this is really how it works. The problems arise when there is a mutation, and after that mutation is passed, then there is a mismatch to the left of your 13th base. So if you imagine just single base substitutions, that's usually not a big problem, and that can be overcome pretty easily with the hybridization conditions. The problems arise when there are deletions or insertions, as they will really displace the hybridization. And when you're using pre-sequencing microarrays, deletions and insertions are really not something they very well identify. It is possible, but it's not recommended to really go after those. The other problem is repeats as well as single base changes in homopolymer stretches. Those are never friendly with any sequencing technology, but microarrays are particularly susceptible to slippage of the hybridization if you have repeats or single insertions in homopolymers or deletions for that matter. Another problem is the large deletions. And here the problem is not as much with identifying them as with designing the probes and the assays. If you know the sequences of the breakpoints, then that's great. You can design your probes and you can really identify those deletions, or at least attempt to do so. If those breakpoints are not known, then microarrays will be really, really difficult to use in such case. Now, I'm not going to spend a lot of time on the next generation sequencing instruments. There is a whole variety of them. The two most commonly used for panels that I mentioned before are Illumina and Ion Torrent. Now, how do the sequencing workflows compare with microarrays versus next generation sequencing? In both cases, we start with genomic DNA, kind of obvious, and we end up with some kind of data analysis, more or less complicated, and a report hopefully telling us what we had in the sample. Now, microarrays are exclusively dependent on PCR amplification and a multiplex PCR at that. If you're designing your own assay and you wish to use Raindens or Haloplex, you can try and experiment with that. I have no first-hand experience with doing this, but the equipment or the method is quite expensive, so you have to think about how that will work for your assay. Once the products are amplified, the pooling or purification needs to be done, and then the remaining steps such as fragmentation, labeling, hybridization or stain and scan follow normal Affymetrix protocols. So there is no magic. You just keep on going. On the other hand, the next generation sequencing requires the genomic DNA first to be fragmented. And then depending on what protocol you're using, you may need to tag those fragments and then amplify with primers that are proprietary, or basically they're not specific for human genomes. So that's a difference between the microarray amplification and this step here. If you're using the target enrichment with amplification, then you may need to, you may tag them after. It really is dependent on your protocol. Once you have tagged big library, then you put it for sequencing, and you end up with some kind of data that you can analyze. So when we, oh God, I really hate sometimes technology. When we actually look at the data, those A's should be all aligned nicely under the G, because we're looking for the G to an A change. The other thing that you also see is those random pink changes, such as C here and a C here and a G here. Those are basically artifacts of sequencing, single changes. But you also see this nice line of C's here that should be all nicely lined up, and it comes under the G, indicated that there is a previously undescribed polymorphism in that position. So it's very clear to identify your mutation and possibly novel polymorphisms. You get all of the sequences read aligned to your reference, and that is called depth or coverage of your sequence. So it's kind of, you see that there is a difference between the microarray, which actually microarray gives you a FASTA file of your sequence, and this type of file that you get from next-gen sequencing, and that format is the FASTQ. Now, I'll talk about the pros and cons of each of the technologies. So microarrays, what is good about microarrays? That you can get a quick turnaround time, and this is just your lab time. 24 to 48 hours, depending on your workflow, how long you want to work during the day, how many shifts you have in the lab. And after that, you obviously have to analyze it, and that will add up a day or a couple days, depending on how you want to process that. The experimental protocol is relatively simple. It's just multiplex PCR, and the differences depending on how complex, how many genes you're amplifying. It will be either really simple, just 1 or 2 tubes, or you will have up to 30 or 32 wells in a plate to amplify. I think the largest I did was about 800 or 900 amplicons in our Charcot-Marie Tooth assay. So that was a lot of amplification. So this protocol wasn't as simple as it sounds. Once your bioinformatic analysis is optimized, the results are coming through really quickly, about 5 to 10 minutes, depending on the speed of your internet on a queue that's on the server. And if you're comparing those arrays to the SNP arrays that you might be familiar with, you actually end up with sequence, unlike the SNP arrays. Now, what are the problems with microarrays? As I mentioned, insertions and deletions are a big issue, and those arrays are not really recommended for it. It definitely requires multiplex PCR for amplification, so primer design is a key issue. You may end up with losing some of the results if your noise is not consistent across the array, and it can happen due to a lot of various unfortunate accidents. If you want to modify your design because your panel has changed or you want to add a gene you just realized you missed or a mutation you missed, depending on your throughput, so your usage of arrays per year, that might be very costly because you may need to pay for mask, and that can add up. It's also not a very rapid change. The other issue, obviously, is that you can only detect what you tiled on the array. You cannot identify completely novel mutation in the region that has not been tiled. Now, the problems we had with the current assays that I worked with were that partly degraded DNA or whole genome amplified DNA didn't work very well. So FFPE sections were not really pretty. And this problem is solvable by altering the multiplex PCR conditions. You have to have shorter amplicons, and then it works really nicely. Now, next generation sequencing, some people think, oh, that's Panacea, we'll be able to do everything we cannot do on Sanger or on microarrays. And that's not quite exactly the truth. The pros, obviously, we can detect insertions and deletions much better. We can detect novel mutations in areas that we would not have tiled on microarrays, but because we're sequencing the entire exon, for example, not just the first 30 bases of it, we can see those mutations coming through. I will talk a little bit later about the reproducibility and incidental findings, but the big plus is the continually decreasing price, as well as ability to actually multiplex more samples per lane. So that's dropping the price down. Now, what are the negatives? As I said, it's not panacea for things that don't work. Next generation sequencing, there are areas for which you will not get full coverage, and this will create the need for additional tests. The sample preparation is more complex. The bioinformatic analysis is more complex, primarily because of the amount of information that you have. The other issue could be the sequence length of the amplicons and the sequence length that is actually of the fragments that are sequenced. And let me give you one example. The Gaucher disease, GBA gene, has a pseudogene, and to get around the problem, we had to amplify large 2 kb amplicons, which obviously for the next generation sequencing will not work very well, or at all. The other problem is kind of in analysis, the risk of overinterpretation, especially with the variants of unknown significance. So just to summarize, I did take time to talk to people that actually used microarray-based assays and then transitioned some of them to next generation or develop new next generation assays. And basically, a few points. The reproducibility has been highlighted to be definitely much better on next-gen sequencing. Turnaround time was, in majority of cases, shorter for microarrays. When it came to data analysis, really the differences were in less accidental findings, but more pathogenic mutations detected. And also, people noted that they have higher confidence in the data that comes through. And that could be related firstly to the data quality, but also to the fact that we are much more used to seeing sequencing results. And sequencing by hybridization is a little difficult for people initially to grasp and then to become really familiar with. When it comes to data quality, definitely this detection of indels, insertions, deletions is the big plus. And the other thing that I was a little surprised was that there are less calls, because when you put all those sequences together, as we've seen aligned to your reference, you can actually eliminate the sequences that are poor or the locations where there is a problem. Validation is kind of pretty similar. Now, I put this nice report here for you just to have a nice break and introduction to the next step. We're all familiar with having your sequence nicely from Sanger, having list of mutations, what really is the nature of the mutation. Sorry, it's in here. And then what's the effect and what is the change in protein. When it comes to both microarray and next-gen sequencing reporting, the critical issue really for us was to identify the QC points, such as experimental QC and QC of the sequences. Because if the sequence is poor or your experiment was really badly done, this has to be repeated, especially in a clinic. You cannot use something that's subpar. Once you have that sequence all nicely and clean, high quality, then independent of whether you're using the microarrays or the next gen, you have to align it to the reference genome. And it will be the difference between aligning, say, 30 bases or 100 bases to aligning the entire genome or entire exome. The very important part is to actually filter out non-pathogenic polymorphisms, to clean it up. And then if you have a list of mutations you're interested in, like for example for carrier screening, we have a list of say 100 mutations we want to detect. We need to identify if they are present or absent, whether they are homozygous or heterozygous. And basically the building of this report will be very much dependent on each lab that has their own preferences. They want to do their own thing, and they have to interact with the bioinformatics team to make sure that everything they need is captured in that report. So now I can kind of imagine the questions out there is, so what should I use? You kind of say, this is bad and this is sometimes better, but sometimes not so good. So if you're considering a sequencing assay for your lab, you really need to answer, I think, those 5 questions, and you will be on your way to a decision. How many tests are you going to do per week? Are you going to do 10 tests, or are you going to do 100 or more tests? If you're going to do a lot of tests, that probably microarrays might be a little challenging, but not impossible. So let's look at the nature of mutations. If you want to just look at the substitution mutations, then you can choose either of the methods. If you want to assay just a couple of genes or a few hundred mutations, you can use probably the microarrays, but if you're looking at a panel of 100 genes, then you definitely need to focus on the next generation sequencing. Some of the labs I've noticed are very attached to the equipment that already is in the lab. We already have the next generation sequencer. Why would we buy an Affymetrix system, or vice versa? So those kind of considerations might be important for some people. Also, the time required for implementation, and I'll kind of talk a little bit about this in a moment. Now, when you think about establishing that assay in your lab, it's not really that different to any other assay you've done before. The first thing is, why do you want to change? Why do you need it? what platform to use, and a few other things. So I'll talk in that kind of order here. Why would you want to introduce a new assay? Because you want to do it faster, better, and cheaper. Sounds really good to me. And the next thing is you want to streamline the tests. You're doing right now tens or hundreds of tests using Sanger sequencing. So if you can put them all in one method, that would take care of a lot of expense and time. You may say, yes, but we'll have to spend time testing, validating, improving. It might not be that useful. But then if you look at your competition and they're already using gene panels, you may reconsider that fact here. You also can provide more comprehensive testing, obviously, if there are 2 more additional genes or 5 more additional genes that can be tested for this particular condition. For example, for cancer, there's more and more genes coming that people want to test. So that might be useful. Also, to meet more of the clinical needs of the customers, that might be more important. Now, when you think about the whole process, developing the new assay is a critical question here. You have a number of possibilities, and depending, first of all, where you are located geographically, whether you are in the US or in other countries, in Europe or in Australia, there will be different regulations as to what can be used. And in many cases, you can just buy off-the-shelf assay, optimize it for your lab, and be on your merry way. In other cases, the labs tend to develop the LDTs because they are worried about the regulatory landscape. And I'll talk about it in a moment too. So once really the critical question here is the content of your assay. What is your application? Are you doing carrier screening? Are you screening for pharmacogenomics? Are you screening for cancer genes, for particular cancers, for breast, ovarian, for stomach cancer, for brain cancers? Those panels will be different. So once you've decided on this part, and before you actually undertake this, you need to also consider what you will need to validate. You will need to validate your platform, make sure that that performs well every time, that it's reliable. Then how will you go about validating the assay itself? What will you need? And then the IT, the pipeline for analysis. Once you have those 2 pieces in place, or at least you know how you're going to solve it, you can start moving ahead with actually doing it, because implementation will follow. As long as you can create those 2 parts here, you are good to go. So choosing the platform here, it's not just next generation versus microarrays. That's the important thing that I've been focusing here. But once you decide either on microarrays or next gen, you need to decide on the instrument type. So even with microarrays, whether you're going with the cartridges or you're going with the strips or plate type assay. With the next generation, obviously, there is more choices. The other issue that was pointed out to me was also the enrichment method is frequently associated with a big expense, so you need to decide what do you want to use, how do you want to go about it. And as I mentioned before, the actual development of an assay is a huge issue for some labs because of the regulatory requirements. They need to meet them. They need to be sure that they can run this assay tomorrow, in 6 months, and in 5 years, that the regulations will not force them to change the assay again or suspend performing the assay because of how it was originally introduced. So off-the-shelf products do not work for every lab. You can work with a different company that will help you with their expertise to develop the LDT. And all of this will be, you have to really look at the time and the cost. And very importantly, is there any documentation from the companies? Now, when you look at if there is validation information from the companies, then it becomes really easy for you to figure out how to go about validating your own assay. What would be the values for your sensitivity and specificity, for your range, for the reportable range and the reference range, and what is the necessary coverage if you're using the next-gen sequencing. They will have some guidelines, so you don't start from scratch. You can actually know, okay, I need this sensitivity or that many genes I can report probably, so let's see if we can get to the maximum, or we'll have to settle on a little less than it's been reported. Obviously, I'm not going to spend enormous amount of time. Everyone that works in a CLIA lab knows that the QC process of the performance specs has to be done, and that will be lab specific and technology specific, but it's critical information. We will come back to the proficiency testing in a little while, because that is for targeted resequencing. It is a problem. Now, one other thing, we've been talking about a reference genome, and there has been, this is still being disputed whether current reference genome we possess for human is really a good genome or not a good genome. I think we need a lot of more of whole genome sequencing before we really be happy with what we have as a reference, but we have to use what we've got. So you can agree or disagree. The other part is the reference materials, and those are critical for development, validation, and ongoing QC. And people tend to ignore this and say, oh yeah, I have this sample that's been sitting in my freezer for 10 years. It has those really precious mutations. I'll just use this to validate or test my new assay. And from experience, I would say bad idea, because the DNA will be degraded. You don't know how well your assay is really working. So you should start with well-characterized samples, and I'll show you in a moment where those samples can come from. So you can have genomic DNA, which is preferred because it's similar to the patient sample. Synthetic DNA is not really representative, and unless it's the only option, it's recommended that it's rather not used. And obviously, for data analysis, you need electronic reference materials. Now, where do the reference templates come from? There has been a lot of work going into discussing the reference materials and making it more uniform across all the labs, because it became obvious we don't have them ready. So Coriell offers both cell lines and purified DNA that has been recommended for certain diseases, actually has been reviewed and characterized by GETRM, and those samples and those lists can be actually obtained and used as your reference. Now, we've developed the assay, we've validated it, and now we're going into implementation, and all of a sudden we realize, gee, we didn't thought about the regulatory thing. So I suggest really strongly to look at this part first, just at least to get yourself familiar with how you will implement it. What is the regulatory requirement? Currently, the FDA does not regulate genetic testing. However, they do regulate direct-to-consumer testing. They don't like that. And I don't think that any self-respected geneticist would like that too, that someone just given their whole genome and said, okay, here you go, figure it out yourself. So there are issues with the regulations, but you do have CLIA and state regulations. You have different countries have different regulations. Now, there are some guidance documents that really need to be looked at, and some of them are very helpful, and they're not redundant, so they really address different aspects. As I said, one thing that all the CLIA labs need is proficiency testing, and basically at the moment there are no formal programs for next generation sequencing, or for that matter for any targeted resequencing. So for microarrays also. There is for expression microarrays, but not for targeted resequencing. So that's up to all of the labs, although guidelines might come up soon. The other, a little slippery slide, is the reporting guidance. And basically, if we have a list of mutations that need to be screened, either recommended by ACMG, that's fine. We're screening, we're reporting. However, if we have incidental findings, it seems that we're bouncing from, oh, we should report them irrespective of the patient's age. We should not report them. So the guidance right now is to report them. However, I wouldn't bank that it won't change. And again, we go into state and country-specific regulations here. Finally, you've got your results. You return the results to the clinician that ordered the test. And according to ACMG, is the ordering clinician is responsible for providing the counseling. So it becomes very clear that there will be need for more education for everyone, how to handle the next generation sequencing, how to explain incidental findings and not overdo it. So in summary, we don't have one method that will solve all of our problems. Both methods have their good sides and bad sides. And targeted sequencing is really going, I think, ahead with a lot of panels for particular use, for oncogenes, for carrier screening, for particular diseases. And I see this as a future really where it's going. Development and validation are complex though. You have to take care of your reference. You have to make sure you have the genome reference, you have the template reference, and you cover the things that aren't covered with alternative tests. And reporting can be really painful at the first development, but once you developed it, it will be very easy to do. Okay, that's the end of the presentation. For those of you that are going to claim the credits, you can go to this link to follow the questions. And I have here questions already coming through and buzzing on my desktop, so I'll go into some of that. The first question that I have here is, with all the assumptions and speculations involved in sequencing technologies, what are the chances of errors in case a sample is subjected again, for sequencing, percent accuracy of such repeat sequencing. Okay, with the next generation sequencing, I don't have hands-on answer for you. From my talking to people that actually done it, they say that it is really high. They didn't really mention the numbers, but 100% of them said that it is much more reliable than microarray repeated resequencing. So I would speculate that it's at least 99%, but that's my guess. And if someone disagrees, please don't yell at me right here. But it will be in very high 90s. On microarrays, that percentage of accuracy with repeated sequencing varied, but it was also very high. I would venture to say that it was at least 95 to 98%, depended on the samples. So samples that had mostly substitutions were 98, 99%. Samples that had either poor initial template quality, and indels in them, those tended to be more error prone. So there are differences not just because of the technology, but also because of the quality of your sample. And I think that's really, really important. The better your sample is, the better your results will be, no matter what is the platform. The second question here is, could you please highlight cost comparison also? So cost for, if you want to buy the microarray sequencing assays, those were varying depending on which assay you were looking at. From, I don't want to lie here, I would say from about $200 per assay upwards. It depended on the size of the assay, so on the array. When we're talking about the next-gen sequencing, I don't have that information right on hand. So, but if you're interested, I can either point you to the people that would know that, or you need to ask labs that are actually running this type of screening right now. Now, the next question, do we need to confirm again by Sanger sequencing after finding a variant by microarray? I had this question coming through a lot, and really the good practice is that yes, you do. Now, obviously, if you find in our carrier screening, for example, we were finding usually about 3, each person would have maybe 3 variants, maybe 4, and you exactly know which ones you have to sequence. So I would say it's not a big deal. If your sample reports like 20, then first of all, look at your QC before jumping into confirming by Sanger. Some labs have said that they're not going to do Sanger confirmation. And I think at the initial validation stage, you have to do it. Once you're really confident in your assay, you may get away without it. But as I said, good policy is to do it. Okay, next question coming here is, how can one use next generation sequencing in disease diagnosis where the disease pathogenesis is not associated with gene mutation? Oh, that one is an interesting question. So if there are, for example, it might be due to differential methylation. or other regulatory aspects. So you can use different type of next-gen sequencing assays, but you would have to exclude first that it is not associated with a gene mutation. So once you excluded that, you can move forward with using, for example, CHIP assay or methylation screen. So there's differential methylation and that's causing the pathogenesis. Okay, next question is, what next generation sequencing platforms are CLIA certified? If none of the next gen sequencers are CLIA certified, can we use the results for clinical decisions? I'm not exactly sure, because if your lab is a CLIA lab, there will be particular recommendations how to make, what do you need to do to certify your sequencers for use in a CLIA lab. So I would recommend talking to the labs that are running the next-gen sequencing, and in Toronto there is a lab that does it's a core lab, and you basically, they are a CLIA certified lab, so all of their instrumentations would be certified. I'm not exactly sure what you're getting here at. So the whole process would have to be adherent to the CLIA regulations, not just the instruments. So check with your provider, or if you have your own instrument, then I would go through and make sure that you have all of the reagents, that there are all the specs are up to scratch. So you create your own rules and make sure that it's just the same as with your Sanger sequencer. Just follow similar rules. I'm pretty sure I'm not off the mark here. But I would check with one of the providers, and if you want to, I would have to look for who that lab is. And I don't see any more questions coming through here. So I would like to thank you everyone for attending this. I hopefully gave you some idea of what is out there and what to do with it. I know I haven't touched on everything, but it gives you a start as to where to go. And this presentation will be available, I think, for download as a PDF, as well as you can listen to this again if you have any questions. I can be found around on LabRoots, and just please be free to contact me through there. Thank you very much.